I'd like to introduce James Ayala. He is a conservation biologist and with 20 years experience working with wildlife. He works at the China Chengdu research base for the giant panda breeding program as the research manager, and he's here to give us insight into this amazing program of conservation that they have in China. Thank you. James.
Great. How do I change my slides? Okay, great. Okay. Before I get started, I would like to thank ISBER for inviting me to speak to you all on behalf of the Chengdu Research Base of Giant Panda Breeding. And I'd also like to apologize on behalf of my director for her inability to attend due to scheduling conflicts. I'm going to give you her presentation and try to do my best job. Thanks a lot, okay? So the Chengdu Research Base of Giant Panda Breeding was founded in 1987. This was a crucial period in the conservation of giant pandas. At this time, there were a lot of factors that led to giant pandas harboring on the brink of extinction. So this base was founded with an effort to save wild giant pandas. The base was founded in 1989, and you can see just 2 years later, it was pretty sparse. We just had a few basic buildings for support and a lot of open fields. And over the past 30 years, we've transformed into one of the largest and most visited tourist attractions in all of China. The scenery itself is worth the cost of admission, but everybody, of course, comes from the pandas. This is easily the best place in the world to see giant pandas. Our population was just started with only 6 giant pandas in 1987 that were malnourished. And since then, we've had 180 litters of giant pandas often twin, and we've had a total of 273 pandas born at our facility. This is the largest captive giant panda breeding facility in the world with a population of about 184 individuals at this moment. That includes pandas in China as well as in our partner institutions around the world. These are just a couple of our cubs. Obviously, they're really cute and they draw a lot of attention. The support the public gives us is amazing based on how cute and amazing these animals are. But we're also still an active and thriving research center. The first building you see here, this is our veterinary hospital, where we conduct our research as well. Our museum, this is our storage banks, where we have the largest semen banked for giant pandas, as well as our genetic banking, and this is our nutrition research center. We also have all the amenities that you'd expect for a tourist center of this degree. We have a large gift shops, various amenities you'd expect. So each year our visitation rate basically increases at about 30%. We were named in 2015 as one of the top 20 tourist destinations in China for international visitors. And in 2015, we were ranked Chengdu was ranked one of the 52 most tourist-friendly cities, in part because of the panda base. We have about 5 million visitors in 2017. These are just a few of our VIP visits that we have. Every year we entertain numerous VIPs. You can see Michelle Obama came, as well as Queen Sofia of Spain, amongst numerous international celebrities. So this is the outlook of the ex situ conservation of giant pandas. There are numerous facilities around the world that house giant pandas. Some of them are partnered with us, and some are partnered with other facilities in China. There are about 6 panda facilities in North America, 5 in Europe, and numerous facilities in Asia, including Thailand, Japan, Singapore, as well as Australia. So that was the global distribution of pandas in facilities. However, the in-situ distribution of pandas is a little bit bleaker, a lot slimmer. They're only endemic to China, of course, and they're only found on these 6 mountain ranges of China. Each one of these red dots is a nature reserve in these ranges. You can see that Chengdu lies kind of around, this is actually a basin, and Chengdu lies just south of this range, making Chengdu the only city in the world that has captive and wild giant pandas. So again, back in the late 70s, early 80s, this was a really critical period for giant pandas. There were two major factors that came together at the same time that caused a tremendous loss of pandas, one being habitat loss and the other being bamboo flowering, which is a natural phenomenon in which the bamboo will flower in an area, and then about 80% of all the bamboo will die off, leaving the pandas with no food. You can see that pandas were in a tremendous crisis at this period. So what initially was done was that pandas were brought into different captive facilities, and the original projection was the population at the time would not be self-sustaining and basically would die off within 60 years. Some of the major issues that pandas faced were related to malnutrition and digestive tract diseases. So we were left with pandas with stunted growth syndrome, completely emaciated, and they had these mucosal feces. So even though we were feeding them, because of poor understanding of panda nutrition, they weren't getting enough nutrition. Also, disease was a major issue. Hemorrhagic enteritis was one of the major killers of giant pandas, as well as canine distemper. Canine distemper is still an issue because in 2015, about 5 pandas died related to canine distemper in Shaanxi province. Rotavirus diarrhea was one of the major killers of young giant pandas. Again, because of poor husbandry and poor hygiene, one panda would get sick, one cub would get sick, and then all of the cubs in the facility would soon become sick after. And it was very characteristic. I don't know if you can see so well, but they had this bloody discharge that would come out, and we were losing panda cubs at alarming rates. Parasitic infection was also a major killer of both pandas in the wild as well as pandas in captivity, and was definitely one of the things that needed to be addressed. On top of all of these health issues and veterinary issues, there are numerous breeding problems with giant pandas. Giant pandas are notorious for their breeding issues in captivity. I assure you that they're fine breeding in the wild. It's only under captive situations that these breeding issues occur. So a few statistics, in the early days of panda conservation, in the ex situ conservation, only 1/3 of sexually mature females actually showed appropriate estrus behavior and would go into estrus. Between 1953 and 1989, only about 22% of the females that did go into estrus and became pregnant actually gave birth. About only 8% of the males were able to sire cubs naturally. So this is kind of really big issues for conserving the species in captivity. In regards to the females that actually did give birth, only about 33% of those cubs were able to survive. One of the major issues, too, with the pandas is that occasionally pandas will twin. About 50% of panda births are actually twins. However, in the wild, pandas will never care for both twins, so one will always die. So for us, in the early days, we had about a 67% survival rate for cubs that were single-born, and about a 19 to 20% survival rate for cubs that were born through twinning. So early on, it was pretty obvious that no single effort would be able to address these challenges, that a multidisciplinary effort would be needed to come into effect. So we combined the in situ conservation with the ex situ conservation, with obviously the in situ conservation being the priority. The first thought was to protect the pandas in the wild, but then have this ex situ population as a safety bank for the pandas. And you can see the different areas that overlap and how that comes into play for their conservation. Beginning with the in situ conservation for giant pandas, the first step, of course, was to protect the natural habitat. So over the course of the years, China has created 67 nature reserves that incapacitate the giant panda habitat. Also, various policies, laws, and regulations were put into effect to protect the pandas, including cutting down on poaching and any sort of new construction in these wild areas. As a result, the giant panda habitat has expanded greatly. So in the 1980s, you had about 1.4 million hectares devoted to giant pandas, and then that has increased gradually to about 2.6 million hectares that are currently available for giant pandas. So we're making progress. This graph basically shows how the panda populations have changed based on the different surveys that have occurred throughout the years. In 1977, the wild population was estimated at just 2,459. This red line represents just about the time that that bamboo flowering occurred, and we had this drastic decrease in the population, where the population basically went down to less than half of its original population. And then you can see gradually the population has increased due to the measures that China has taken. Currently, the last estimate was 1,864, and that's based off of a 2015 survey. So as the giant pandas have improved and the habitat has increased, IUCN took the recent decision to downgrade giant pandas from endangered to vulnerable. This is kind of a dangerous thing because there are many factors that still can affect population of giant pandas. They're very specific animals with their diets, so they're especially vulnerable to climate change. So it's predicted that about 35% of the current habitat available to pandas can reduce greatly in the next 80 or so years. So they're still not very much so in the clear as we'd like to think. So that was the current update for the in situ population. But the response that was taken for the ex situ population was this creation of these different research centers. One being the Chengdu Research Base of Giant Panda Breeding, where I am, and this is our current entrance. We also have another facility in Dujiangyan, which is in the natural habitat of giant pandas. Several other facilities were created as well, such as the China Giant Panda Conservation Center in Wolong, the Shaanxi Rare Wildlife Conservation and Research Center in Shaanxi Province, and Beijing Zoo. Beijing Zoo is basically the center for the Chinese zoos that house giant pandas, but there are numerous zoos now that house giant pandas in China. So we began slowly with the formation of these facilities to address the technical issues that we were having. It seems pretty apparent and obvious now, but at the time it was quite difficult to understand. But pandas need bamboo. So the major step that we had was switching their diet to almost 90 to 100% of the natural bamboo that they would eat in the wild. This includes not just the bamboo, but following the appropriate season and type of bamboo available per year. So for example, roots would be available in the spring during the mating season, shoots available at different times of the year, and then in the winter when they eat mature bamboo, getting the leaves to them. This drastically in itself increased and improved the health of the giant pandas. We began to address different issues as far as disease control through a partnership with different zoos and collaborations in the States. We began using different vaccines to treat canine distemper, to vaccinate against canine distemper. Currently, we use a malarial ferret vaccine. It's very interesting. The vaccine was designed for black-footed ferrets, but it works very well for other carnivores like giant pandas. We also have done a good deal of work detecting and preventing the rotavirus, so our animals no longer suffer from this disease, and we're able to treat it much rapidly if it ever does occur again. Parasite control, this is still something that we're working on as new parasites are being discovered. And again, this is something that we're concerned with global warming and the increase of parasites, but we were able to identify about 22 different species of endo and ectoparasites that infest giant pandas and come up with different treatment regimes for them. The problem of reproduction, this is an area that's actually my field of research. It's still something that we're working on, but we've made great strides. We've done a lot of work with panda endocrinology, so we can understand the female estrus cycle. So for example, just looking at this, you can see your estrogen will increase. This is the ideal time to do mating. There's about a 3-day window in which their estrogen is elevated that they'd be interested in reproducing. So this is the time that we would do natural breeding and artificial insemination. And then you can see as estrogen decreases and progesterone increases, this will signify that she'll give birth shortly. So this is very useful for us in managing the giant pandas. We've also done a great deal of work with artificial insemination and the timing of artificial insemination. We've also addressed the issue with giant panda twins. Through training and a lot of the hands-on work we do with the pandas, we've come up with a method that will allow us to swap cubs from one female to another. So in the event a female has twins, we can take away one of her babies and kind of rotate them around our group of lactating females to ensure all of the cubs get the appropriate nutrition, especially the colostrum that's needed for giant pandas. We are also able to milk the giant pandas to make sure that, for example, if a female in the US isn't lactating, we can send milk from China to that animal and make sure that they get the appropriate nutrition. Another issue with pandas, and something really unique to their biology, is pandas give birth to the most underdeveloped cubs. All panda cubs are considered premature based on their development. So they're born deaf, blind, they're not able to thermoregulate, they can't defecate. So usually a normal panda weight will be about 1/1000th of their mother's. So you'd have a 100 kg mom giving birth to a 100-gram baby. In some extreme cases like this one, this cub was born, you can see how tiny it is. This is a 100-gram panda, this one was only 51 grams. By using different methods that we basically borrowed from working with human premature babies, we were able to ensure the survival of these cubs. So this is no longer a major issue for us. 51-gram cub is quite tiny. The fact that this is now a huge breeding male was one of our major milestones in their conservation. As a result, you can see the panda cub survivorship has increased greatly. I'm not so sure if I trust this data, and I apologize for being in Chinese. This blue is the number of singletons that were born, and the red is the actual number that survived. The purple is the number of twins that were born, and this is the number that actually survived during this period. So you can see that more than half of the cubs during this period were dying off. And now we come to here, and we have about an 80% survival rate of the cubs. Okay? So we've been doing, an improved way in breeding our cubs. As far as genetic management, this is just a brief overview and timeline for some of the milestones we've achieved with genetic management. In the 1980s, the first protein isomer markers were created. In 1990s, DNA fingerprinting technology was implemented for giant pandas. In 1996, it was very interesting. We had 8 teams launch different R&D microsatellite markers for the giant pandas. And then in 2001, we had a large-scale application of paternity data for giant pandas. The reason paternity data is very important is because we often use artificial insemination in conjunction with natural mating. So we may have semen from different males being used, and determining which one is the father, is obviously very important for the genetic management of the giant pandas. So during the last 16 years of research, 11 giant panda microsatellite loci data sets were structured, obtaining the genotyping standards for 68 giant panda alleles. Now this is our standard method for pairing giant pandas for breeding. Every year prior to the breeding, we establish which animals will be mated, and we have priority listings for the most ideal individuals to be used. Because of this, we've had improvement in our genetic diversity. So we've went from 9.688 up to 0.97, which is a small increase, but it is moving in the right direction and something that we're working towards improving. So initially, when we first started breeding giant pandas, a number of 500 was established as the ideal number for us to maintain a self-sustaining population. And you can see over the years that we've reached that number. We hit that number around 2015, but we're still planning on continuing to breed giant pandas to further improve the genetic diversity. As I mentioned before, too, in Shaanxi province, there was a loss of pandas due to CDV, canine sempervirus. So we're also trying to make sure that we can safeguard these animals even as the population grows. This is where you guys all come into effect. We do have banking for our samples. We have our sperm banks, somatic cell bank, stem cell bank, and a tissue samples library. We're very much so interested in improving our storage capabilities. You can't have good research without good samples. So I would, be very grateful to speak to some of you after about different ways that we can do that. One of the most important aspects of our facility and any conservation program is the formation of public education. All of the work that we're doing in trying to conserve this species is useless if we do not have any of the public support that goes along with it. So we were the first facility in Sichuan province to establish a conservation education program. One of the main components is actually going to the local schools located around the nature reserves and teaching the students how to improve their wildlife, respect wildlife, and foster this idea of stewardship so that the next generation can continue the work of conservation that we're doing. So not only do we have a dedicated education facility at our base, we also travel around China and foster this notion. We're not in the clear. There's still many threats that affect giant pandas. The population is growing, but it's still very fragmented. So although it's around 2,000 individuals, they're actually broken up into 33 different groups. 22 of them are still at extinction risk, and about 18 of them, in some of these smaller pockets, you see, there are actually less than 10 pandas in those groups. So if there's a bamboo die-off or any change, we can lose that population pretty much at any moment. To address this, the Chinese government has recently come up with this idea of creating a giant panda national park. The goal for this park would be enhanced habitat connectivity, creating land bridges and so forth to connect these reserves. It also solves the issue of multiple management. Currently, each reserve is governed by the province that they're in, and they have very different regulations. But this would impact, only one regulation amongst all of the reserves. And it also will allow us to establish new administration. The current plan, again, is to have 2.71 million hectares devoted towards giant panda habitat, and this is 3 times the size of Yellowstone National Park. We're also doing different reintroduction and restocking programs. Currently, the Wolong Panda Base, which is the China Conservation Program, they're working on using the mother with young wild training way. This is a method in which a female will be released with her young cub into the wild. Cao Cao was one cub successfully reintroduced. And then Shangjiang was a panda that was a female that was released into the wild and became inseminated by a male panda and then was brought back into captivity. At the Chengdu Research Base of Giant Panda Breeding, we're working on using an assisted soft release program in which pandas will be paired with a human and they'll go out for, basically walks through the reserve to acclimate them and habituate them to the wild area. And then they would disperse from that person the natural way that a mother would. It's more of a natural and slowly progressive a way to release an animal. The benefit of that, too, is that we can get close to our pandas and change the collars on them so we can ensure that we can monitor them while they're released. This is actually my director, Horong, working in the field using radio telemetry to follow our bears. Something we definitely need to do is we need to strengthen our banking. We need to improve the way we store our genome samples, our genomic samples. This is something that we need to look to towards the future. And again, if anyone would be interested in cooperating with us, we'd be very grateful for that. The wildlife, pandas don't just exist in their habitat alone as a single species, and to think that you can conserve one species and not others is quite a foolish notion. So we began working with other organizations and also on our own to start looking at conservation projects for different animals, especially Chinese animals, such as the Sichuan snub-nosed monkey, red pandas, the South China tiger, soft-shelled crab. This is the Chinese mono, red-crested crane, and snow leopards. Also, you can never have enough education, and that's something that we do very well. We're very open with the public. We have very many meetings. We have different media groups coming and keeping the public up to date with our animals, but it's always a challenge to maintain this. There's often an issue between welfare and conservation, so having clean, open conversations with the public is something that we strive to do and keep everything clear with the public as far as what's going on with our animals. So that's it for my presentation. I'm a little quick, so we have time for your questions. Yep. You're welcome.
Can you come up to the mic to ask questions? There we go. And please introduce yourself first. Thank you.
Sure. Can you hear me? So my name is Marta. I'm from the Cornell Veterinary Biobank. Your talk was very interesting. I just wanted to know if you've started use of those samples that you've been banking. If you've distributed them and,
Can you repeat that? I'm sorry?
if you've started to use the samples that you've collected.
Which specific samples would you refer to?
You presented your biobanking efforts into the genomic.
Yes, yeah, we've been using our samples. There are numerous publications. I'm not the geneticist, but we do have several publications. The point I wanted to raise, though, is that I do think our sample storage method is a bit outdated, and we definitely can use some help with that. Yeah.
Thank
Sure.
you.
So I had a question myself. You said that rotavirus is an issue or is a solved issue. Is there any attempt to adapt the rotavirus vaccine in humans to potentially for pandas? Do you know that?
That's a very good question, but I'm not sure if I can answer that. Yeah, we need to speak to the vet. Yeah.
I would think that
Yeah.
that might be a...
One of the major issues with rotavirus was initially a husbandry issue with just poor hygiene. And I think that just using hygiene measures in itself was able to do a lot of well in that area.
Great.
Virginia Lavolsi from the University of Pennsylvania. I'm just curious as to what, if anything, can be done about the problem with the bamboo diseases that are destroying the bamboo. Is there any focus on that?
The problem is it's not a disease, it's a natural phenomena. As far as we understand, it appears to be cyclic, where basically when you see a clump of bamboo, it's all one individual life form, and then they all flower and then die off afterwards, and then they will regrow. But the problem is during the period of flowering and death and regrowth, the pandas are left without food. But some research is being done into that. But we're panda specialists, not bamboo specialists. But trying to predict, you're right, trying to predict when the bamboo die-off can occur would definitely take things in a great, go in the right direction. Yeah.
Any other questions? Oh, here we go.
My name's Micah. I'm from a central lab in Ohio. And you mentioned global warming a couple of times impacting the population of the panda. I was wondering how the urgency with, repopulating the panda impacts climate change policy, maybe. It seems like the industrialization in China would have some impact on your ability to do your work. So
Yeah,
there's a relationship there.
Yeah, climate change is a major problem, and I didn't mention this during my talk, but pandas are animals that prefer high altitude, and they prefer low temperatures. So as climate change increases, the pandas will have to move out of the reserves or move higher up into the mountains, which may not make the appropriate bamboo available. As far as the effects, China is a rapidly growing market. I mean, it's a developing nation. It's very interesting, though. Like you see in the city where I live, there's been a strong push to being green in an attempt to reduce the effects of climate change. So you'll see a lot of green vehicles, a lot more electric cars, and a lot more electric trucks than you would see in the States. But definitely, it's this is the situation. You have this growing population, this rapidly growing economy, and the impact it's having on the environment. So we're not sure how that's going to go. Yeah.
So I'm positive there are ISBER folks here that would love to partner with you to help you.
Sure, we're
build
very
out your
open
biobank.
to collaborations, and I think too for the Shanghai meeting, we'll definitely make a much stronger appearance. Hopefully.
Perfect.
Yeah, great.
Great. Thank you
You're
very
welcome.
much.
Sure. Bye-bye.
Next, we'd like to invite Dr. Sarah Lasky to the stage. Dr. Lasky joined 23andMe in 2016. She works as a research scientist on a small team focused on developing the next generation of 23andMe's consumer health platform. This team works to keep 23andMe at the forefront of science and technology, driving the development of interventional studies and predictive models for human health that utilize genetics, lifestyle, and environment. Dr. Lasky earned her PhD in biomedical engineering from Johns Hopkins University. Welcome, Sarah.
Thank you so much, and I want to thank ISBER and the organizers for giving me the opportunity to present to you today. I'm here to speak about the amazing and unique 23andMe cohort and some of the great science we've been able to do with the data that they've provided us. I always like to begin with the mission statement of 23andMe. So our mission as a company is to help people access, understand, and benefit from the human genome. And on the research team, we're focused mostly on the third facet of this mission. Our mission is to help people benefit from the human genome, and we have 3 overarching research goals that get at that underlying mission. So the first of those is that we hope to understand the biological basis and specifically the genetic basis of human health and disease. We want to use this understanding to develop therapeutics to treat diseases and help humans who are living with disease. And we also hope to use that understanding to delay the onset and prevent the development of disease. So those of you who have heard of 23andMe probably have heard of us as a consumer product. 23andMe is a direct-to-consumer genetic testing company, and this is an image of a 23andMe kit. So the way that this service works from the perspective of a consumer is that you log online, you order a kit, you receive a tube in the mail, you spit in the tube, and then you send your saliva back to us. We analyze the DNA in that saliva and ultimately provide you with personalized genetic reports. So this is how we collect genetic information from the participants in our cohort. And the technology that we use to measure genetics is something called genotyping. So this is often contrasted with sequencing. In the case of sequencing, you measure every single position in the human genome effectively, whereas with genotyping, you measure intentionally specific locations of known variation from person to person. So in this toy example, you can see there are 3 people whose DNA is identical over this stretch of sequence almost entirely, and this is representative of how something like 99.5% of my DNA is identical to the DNA of everyone in this room and everyone on this planet. So, but you can see there's one position where the pink person has a T and the green and blue people have A's. So this is a position of known variation. This type of genetic variant is called a single nucleotide polymorphism, or a SNP, and these are the kinds of genetic variants that we measure with our genotyping array. So the array that we use is a customized version of the Illumina global sequencing array, and it measures about 600,000 of these single nucleotide polymorphisms. And in addition to that, using an algorithm called imputation, we can infer up from those 600,000 variants to about 10 million different genetic variants. So ultimately, from our entire cohort, we have something like genetic information at about 10 million positions in the human genome. This is remarkable. This is quite a lot of data. But what makes the 23andMe database really powerful and unique is not just this wealth of genetic information, but the fact that our genetic data is paired with phenotypic data. And so for the purposes of this discussion, I'm using the word phenotype very broadly. When I talk about phenotype, really I mean any information about a person that's not their genetic information. And so we collect phenotype data through a process of online surveys where we just ask people. We ask them about themselves. We ask them, how's your medical history? How's your lifestyle? How's your environment? How much do you eat and sleep and exercise? And we just ask them questions online, and they answer. And of course, this process of research is all IRB approved. Customers opt in to participate in research. It's not mandatory. And of course, they can withdraw their consent anytime after they provide it. And most importantly, the data that we publish are published in an aggregated and anonymized format. And so no individual could ever be identified from data published by 23andMe. So the final point here I want to make is the advantages of this online research platform. It's really easy for participants to provide data, phenotypic data in particular, because they're just answering multiple choice questions on their phone or on their computer. They can do it at any time of the day. They can do it from anywhere in the world. And one other thing that's really remarkable about this research paradigm is that each research participant can be part of many different studies. And so this is really different when you contrast it to sort of a traditional research paradigm where every single medical condition you want to study, you have to go out and recruit an entire cohort for that study. In the case of our very large cohort, for any condition or trait that you'd like to study, you can divide the cohort into cases and controls based on that particular condition that you're interested in looking at. So we take all of this genetic and phenotypic data and we use it to search for genetic associations. And when I talk about genetic associations, what I mean are genetic variants that are associated with a condition or a trait significantly are different between different populations. So the example that I'll talk about here is a genome-wide association study, or GWAS, looking for genetic variants associated with depression. So we separate our cohort, like I said, into cases and controls. So the cases are people with depression, represented by the sad pink face on the right. And the controls are people who have never been diagnosed with depression, represented by the happy yellow face on the left. And what we do is we just go through those 10 million genetic variants one by one, looking for genetic variants that are found that are significantly different between those populations of cases and controls. So this is what the results from a GWAS typically look like. This is called a Manhattan plot. It's supposed to look like the Manhattan skyline. And what you see is, along the x-axis, just position in the human genome from start to end, broken into chromosomes. And on the y-axis is a measure of statistical significance. So negative log p-value, so higher means more significant. And the yellow line at the middle, it's at a p-value of 5 times 10 to the negative 8th. This is the standard cutoff for statistical significance after Bonferroni correction when you're measuring this many variants. So each point on this plot is an individual genetic variant, and you're looking at the association between that variant and the phenotype being studied. And in the case of this Manhattan plot, this is a Manhattan plot for tongue curling, the ability to curl your tongue. And you can see that there are 2 regions of the genome that we found that are significantly associated with the ability to curl your tongue in chromosomes 17 and 6. So this is how we identify genetic associations. And when you put all of this together, we have 5 million customers, over 5 million customers. 80% of our customers consent to participate in research, and about 3/4 of them take at least one survey answering these phenotypic questions. Altogether, these 5 million people have contributed over 1 billion phenotypic data points. That's billion with a B. They've answered that many survey questions. And we have used the data they've provided to publish 100 peer-reviewed research publications and counting. So if you want to take a look at the kinds of things we study on the research team at 23andMe, you can go to 23andme.com/publications and take a look at these publications. And this brings me back to those big 3 overarching research team goals. The first of which is to understand the genetic basis underlying human health and disease. And that's what we do with these genome-wide association studies and with these publications, is we probe into that genetic basis of health and disease. For this second goal of treatment, I'm not going to talk about it today, but we do have an entire therapeutics division that is devoted to using these genetic findings to develop new therapeutics. But what I want to focus on for the rest of my presentation today is goal number 3, prevention. And I'm going to dive a little deeper into what I mean when I talk about using data to prevent disease. So what we want to do here is we want to begin with the genetic and phenotypic data that I've described. Given that genetic and phenotypic data for an individual, can we predict that individual person's likelihood of developing a given disease, and then suggest personalized actions that that person can take to mitigate their disease risk, ultimately delaying the onset or even preventing entirely the development of disease? And I know this is pretty high level and abstract, so I'm going to dive a little deeper even into a case study on weight. and specifically healthy and unhealthy weight. So it'll be no surprise to anyone in this room when I say that weight is increasing across the Western world, especially here. This is a plot of obesity rates in the United States beginning in 1960 and going through the mid-2010s. And what you can see is obesity and extreme obesity are on the rise in both women and men with no end in sight. And of course, this is a dramatic public health problem because overweight and obesity are major risk factors for a whole host of diseases, including cardiovascular disease, type 2 diabetes, high blood pressure, breathing problems, including sleep apnea, and even some kinds of cancer. So the problem of overweight and obesity in America is a big one, and across the world. What do we do then to maintain a healthy weight and to lose weight when we're not at a healthy weight? We diet. But of course, not all diets work equally well for all people. So the data I'm showing here come from a paper that was published in JAMA earlier this year, where they did a randomized controlled trial comparing a healthy low-fat diet and a healthy low-carb diet. And so across the x-axis are each of the participants in the study, and on the y-axis is how much weight those people gained and lost over the course of the 12-month study. And there are two features of these data that I want to point out and emphasize. The first is that when you step back and look at it, these two charts look pretty similar. There's no one obvious right answer for what is the right diet if I want to lose weight. And the second thing I want to point out is the heterogeneity from person to person. There's a tremendous amount of inter-individual variation here, where some people have lost 20 or 30 kilograms, which is a ton, it's not a ton of weight literally, but it's a lot of weight. And some people have gained up to 10 kilograms, also a lot of weight when you're on a diet trying to lose weight. So this is the kind of question we hope to be able to answer in a personalized way. What is the right diet for me based on my genetics and my preferences? What is the right diet if I want to lose weight? So this brings us back to the path to prevention in the context of obesity and overweight. Given the data for a particular person, can we predict their propensity to be overweight, to weigh a lot or a little? And then can we suggest personalized actions for them to manage their weight, thereby preventing or delaying the onset of these diseases that are driven by obesity? So this is exactly what we've done. We've already talked about the data, the genetic and phenotypic data. What I want to zoom in now on is the next step in this process, the prediction process. And I want to talk about how we use data to predict someone's underlying risk for disease. And the technology that we use to do this is called polygenic risk scores. You'll also hear them described as genetic risk scores. And this technology begins with the algorithm that I described earlier, a GWAS. So in this case, we performed a GWAS on BMI. So we took about 600,000 participants of European descent for whom we had genetic and BMI data, and we ran a GWAS, and we found over 300 genetic variants associated with weight, or BMI in this case. And then we put those 300 genetic variants into a model. So you see here on the left, GRS stands for genetic risk score, and this genetic risk score is someone's risk for BMI, for high or low BMI, based on their genetics. It's a linear combination of their genotype at these 300 genetic variants and age and sex. And what we see is that this polygenic risk score does a great job of segregating people by their actual BMI. So the data you're looking at here are in an out-of-sample testing cohort, so people this model was not trained on, and we've divided these people into 20 bins based on their genetic score, their predicted BMI. So low predicted BMI on the left, high predicted BMI on the right. And on the y-axis is the actual BMI of these people that they've provided to us. And you can see there's this nice monotonic relationship where people with lower predicted BMI have lower actual BMI and vice versa. And so this is really exciting. It means that we can actually get at the genetic underpinnings of weight. And we can see that this model performs exceptionally well compared to other models that have been produced in the literature to do something similar. So the best model, sort of the gold standard at the time this was made, was this model by Locke et al., which is a massive meta-analysis of several GWAS for BMI, and they managed to explain 2.7% of the variance in BMI. The model I just described to you, using genetics alone, can explain 5% variance, 5% of BMI variance. So that's almost double the next best model out there. So now that we have this great model for predicting genetic propensity to weight, what do we do with it? We give it back to our customers. So this is the virtuous cycle of product and research and development at 23andMe. We begin with the consumer product I described, where participants spit in a tube and answer questions to give us genetic and phenotypic data. Those data drive our research, which I've also described, and then when we build models, we put them right back into the product to give them back to the customers who made them possible in the first place. So the way that this looks in the 23andMe product today, the model that I've just described to you, is our genetic weight report. So this is a real screenshot from my genetic weight report, which says my genes predispose me to weigh about 12% less than average. Great, that's pretty cool. That's the result of the model for me, but there's one more really interesting thing that we've been able to do in this report, and that's taking that genetic score and looking for gene by environment interactions. So what that means is places where my genetics impact the way that my lifestyle decisions are going to affect my weight. So these gene by environment interactions, you can see, these results are based entirely on my genetics, and they tell me, for someone with my genetics, what are the lifestyle actions that are likely to have the biggest impact on my weight? And this is really exciting, this is really interesting, that this is personalized, because it's based on my genetics, it's personal to me. So if you look at someone else's report, this is a screenshot of the report from a coworker of mine, he has completely different recommendations. So his top recommendation is that he needs to avoid fast food. Mine is that I should limit my red meat intake. It looks like exercise is more important for my coworker, where getting a good night's sleep is more important for me. So we have these kinds of insights. This is the state of the genetic weight report as it exists in the 23andMe product today. And if you are a 23andMe customer, you can go into your genetic weight report and see these recommendations right now. But what I want to talk about moving forward is where we're going next. What are we going to do on top of this? How do we improve this work? So number one on the left here, we are starting to collect new kinds of data. So the logos that you see here represent our cognitive tools. These are almost like computer games that you play, and they measure things like your response time and your memory. And the goal of these tools is to measure, while people play these games, their cognitive function, including cognitive decline with age, especially in the context of dementia. So understanding how cognitive function declines as we age. Number two, in the middle, new kinds of studies. The logo here is for our weight loss intervention study, which I'll talk a little more about in a few slides. And number three, more powerful models. So the model that I described to you to predict BMI using genetics, while it is the most powerful model of its kind out there, it's not the best we can do. As I said, all of the different genetic variants enter into that model linearly. We have also started working on more complex model architectures, machine learning algorithms. This little icon here is a neural net. We have used neural nets to predict phenotypes, including but not limited to BMI. I'm not going to talk about that work today, but what I am going to talk a little bit more about is how we can use other factors in addition to that genetic risk score to get a better prediction. So what I showed you before was that when you predict BMI as a function of genetics alone, so GRS is that genetic risk score, you can explain 5% of the variation in BMI. But we all know genetics is not the only thing that drives how much you weigh. There are a lot of decisions we make every day that impact our weight, how we eat, how we sleep, how we exercise, and on and on and on. When we predict BMI as a function of these lifestyle behaviors, all of a sudden we can predict a whole lot more variance. And of course, if both of these work so well, why stop there? When we put genetics and phenotypes into a model together, we get the best performance of all. So the third bar on this chart shows what happens when you predict BMI using the combination of genetics and phenotypes, just entering into the model linearly, like I've described already. The bar on the right is also a linear model with genetics and phenotype, but it includes interaction terms between genetics and each of those lifestyle factors. So what this goes to show is that you can dramatically improve model performance by including phenotypic factors, by including more phenotypic factors, and by increasing the model complexity so that it can actually capture some of the real complexity of what goes into driving how much we weigh. And I want to emphasize, I've talked about all of this in the context of BMI, but of course this methodology applies to any disease or trait even that you like. You can use this to predict people's risk of cardiovascular disease or diabetes or what have you. So, going back to our path to prediction, we've described the data, the genetic and phenotypic data. We've described the genetic and phenotypic risk models that we can use to go from those data to predictions of people's underlying risk for disease. Now let's talk about that next step, getting from prediction to action. So this is actually especially complicated by the kind of data you use. So everything I've talked about so far, I've described a cross-sectional data set where we ask people once, what diseases have you been diagnosed with? Tell us about your lifestyle. Tell us about yourself. And this is great. This provides an enormous amount of data, but it's also limited. So a typical, and it's limited because of this classic problem where correlation, you can identify correlations in that data set, but they don't imply causal relationships. So a typical example of this is when we tried to use this methodology that I've described to build a model to predict what are the lifestyle decisions, the lifestyle factors that drive the development of type 2 diabetes. So we have a model. We put in all of the phenotypes, the behaviors in the environment, and we use it to predict type 2 diabetes. And what do we see? The lifestyle behavior or one of the lifestyle behaviors that is most predictive of whether a person has been diagnosed with type 2 diabetes is if they limit sugar and simple carbs in their diet. So if you're really naive, you look at that and go, oh, cutting sugar in your diet leads to diabetes, which is, of course, completely opposite. The truth is that once someone has been diagnosed with diabetes, then they dramatically reduce the amount of sugar in their diet. So this is a classic problem of any cross-sectional data set. And at 23andMe, we are now pushing really hard to collect longitudinal data. And again, with our data collection paradigm, this is really easy. You just give somebody a survey, and then a year later, you give them the same survey. And you can see how people's lifestyle changes over time. So collecting longitudinal data allows you to identify exposures over time, so you can see, how does the cumulative effect of, for example, smoking on your disease risk over time. It allows you to mitigate cohort effects, where you can look at major differences between 20-year-olds and 90-year-olds and ask, okay, well, is that a difference that's driven by age, or is it driven by the environments that these two groups have experienced over the course of their lives? So again, we're really excited about and pushing on getting as much longitudinal data as we can. The other thing that we're pushing into now and very excited about is interventional studies, because of course, the gold standard for demonstrating a causal relationship is to run a randomized controlled clinical trial. So I want to talk a little bit now about the weight loss intervention study at 23andMe, which is our second ever interventional study, but certainly not the last. The goal of this study was to discover and validate genetic variants associated with weight loss and also associated with weight loss success on specific diets. So, getting back to this question that I described earlier of which diet is right for me based on my genetics. So in this study, there are 3 intervention arms. There is a low-carb arm, a high-fiber, low-fat arm, and a physical activity arm where people did not make any dietary changes. And so this study began back in January. We enrolled people in the first couple weeks of January in that sort of New Year's resolution time. We enrolled a bunch of people, and then we put them into this double randomized trial design. So first, we randomized people to either a random arm or a personal choice arm. And then participants who were randomized to the randomized arm were randomized a second time to one of these 3 intervention arms. And the participants on the personal choice arm got to pick their choice of these 3 interventions. And this trial design allows us to understand the effect of personal choice on things like diet adherence and ultimately diet success. So the intervention period lasted for 12 weeks. Over the course of those 12 weeks, once a week, we sent out a survey asking people, how's your diet going? What have you been eating? How much weight have you lost? We've just finished now collecting all of the data, and we are crunching the numbers as we speak. I don't have any data to present from this study, but we are also planning long-term follow-up, so 6 and 12 months after the survey, or after the study ended. So going back to our path to prevention, we are going to use these kinds of interventional studies to really derive those causal relationships and understand which actions will drive lowering of disease risk. The third and final step in this process, going from action to prevention, of course is going to require longer-term follow-up because it takes a long time, it takes years to understand how behavioral changes impact the development of these diseases that develop over the course of decades. But I can say that at 23andMe, we are really excited today to be collecting the data that will drive the models of tomorrow and ultimately get us to that goal of a future where preventable diseases are actually prevented. So with that, I want to thank you all for your attention. I want to thank the millions of 23andMe research participants who made this research possible. Really, nothing we do could happen without them. So the biggest thanks to them. Just a few quick acknowledgements. My team is the health R&D team on the top. The product R&D team below does a lot of the prediction modeling that I described. And a few key players on the research team who made the weight loss intervention study happen. But I also want to emphasize that this is like the short list, this is the highlights. Really, the work that I've described here, it was done by hundreds of different people at 23andMe on all these different teams, and I want to thank every one of them, and thank you guys again.
We do have several minutes for questions. I know you probably have quite a few. Please do come to the microphone, and don't forget to introduce yourself. Let us hear your name and the institution or the organization you're representing.
Hi, I'm Elizabeth Main. I'm representing the H3 Africa Biorepository, so that's a close cousin in some ways to what you're doing. So I have a couple of questions and a comment. So one of the things that I've seen circulating on the ISBER forum has been whether we should consider biological data as a biospecimen. I noted in your talk you didn't speak about biospecimens, so I was just wondering, do you collect these samples? Do you store these samples? That's my first question. The second question is obviously your model is predicated on people signing up for it, which means there's going to be some sampling bias, and whether you had thought of any ways to reduce that. And then obviously the third way is it's great to collect longitudinal data when you're doing interventional trials. You didn't mention epigenetics at all, and I was just wondering whether you'd looked into reviewing your genetic data at any point. Thanks.
Thank you for the questions. Okay, let's see. Number one, yes, we collect specimens. We give participants the opportunity to decide whether we hold on to their saliva or not. I don't actually know the proportion of people, but a large proportion of people do consent to have us hold on to their saliva, and then we can use that for further testing. So we have a number of sequencing initiatives where we go back to those original specimens and sequence full genomes, which gives a much greater depth of information than the original genotyping technology I described. The second question you asked was about sampling bias, and that, yes, we think about a lot. That's a huge issue and a huge point of concern, especially when you want to make medically relevant discoveries. You need to make sure that those discoveries are applicable and extensible and generalizable to a wider population. So we do a lot of testing. When we build these models, we then go back and identify how they perform in different populations. So for example, the model that I described where we did the initial genetic discovery in a cohort of European participants in particular, we do go back and the genetic reports that are surfaced to customers are not just based on that European model, they're recalibrated in different populations. So participants who are not of European descent receive a different result than they would if they were of European descent. But it's something that we're really working on. It's something I'm personally very passionate about, paying attention to the cohort of people that we're using to build models and making sure that those models are extensible to other populations. Your third question, what was the third question? Epigenetics, yes, okay. So I'm gonna extend my answer to talk about not just epigenetics, but all sorts of other stuff. So there's gene expression, there's epigenetics, there's microbiome, there are all kinds of different spatially and temporally aspects of human biology that change. So what's really great about genetics is you can measure it once and you know everything you're ever gonna need to know. Whereas epigenetics, you have differences spatially and temporally over the course of someone's life in different tissues, and the same is true of gene expression, and also very, very excited about microbiome. So these are things that we are constantly thinking about how we can get more data, what kinds of data we can get, and what kinds of data would drive our models. Right now, we're not working on any epigenetic data in particular, but we have talked to other groups. It's something we hear about and it's something we think about. And in general, these different kinds of data collection, collecting different kinds of data, we're definitely interested in because it's always going to make the models better if we can incorporate it.
We could go to this side and then come back to Kathy and then alternate.
So, hi, very nice talk. I'm Puching from GlaxoSmithKline. Just to have a question about the quality of data. I think that's everything in terms of research. Sounds like a lot of your data are collected through self-survey, and I'm just wondering how the quality is and how does that impact the result of your research?
Yep, that's a question we get a lot too. My answer is that the data from self-report tend to be of much higher quality than you would expect. Obviously, there are some exceptions. There are specific things that you know when you ask people you're going to get biased data back. But generally, the proof is in the pudding. You can see from our publication record that this database is the most powerful database for genetic discovery in the world, not despite the quality of our data, but because of the quality of our data. People, when you ask, have you been diagnosed by a doctor with this disease, people tend to remember. And you get pretty high quality data back, and you get pretty high quality research results. It may not be the clinical standard of assaying these disease diagnoses, but we more than make up for that with sample size and ultimately have the power to detect more genetic relationships than you would with a much more heavily curated but much stronger database.
Okay, thanks.
We are actually at our time limit. If we could get a couple of just super quick questions that might require a quick response. How long will you be here? For a few minutes? I know you're not staying very long.
Yeah, I'll be here.
Maybe you could speak, those of you who still have questions, speak with Sarah later. We don't mean to put you off. We just want to keep the session moving. Let's have at least one more question if we could.
Just really quickly, Sean Westaway from Kaiser Permanente Center for Health Research. I was looking at one of you have a great cohort, a huge cohort that's to be congratulated. In your example where you showed the phenotype alone and the genotype alone and then the phenotype of the genotype, can you comment on how significant is that difference between genotype alone and genotype and phenotype and then just using the phenotype? Thanks.
Thank you. So obviously you could see there's more variance explained by phenotype alone than by genotype alone, but I would say that the genetic risk score as a predictor in that model is on par in terms of effect size with the strongest environmental predictor. So all of the environmental predictors together give you more information than the genetics alone, but the genetics alone enters into that model as one of the biggest predictors. Thank you.
Thank you. Help us in thanking Sarah for her wonderful talk. Thank you.
And our last speaker of our session is Michael Lang. Michael's a researcher at the Center for Genomics and Policy at McGill University, Faculty of Medicine. He works primarily on ethics and legal issues associated with digital health, with particular focus on smartphone applications in clinical research settings. He's written on professional and civil liability, research consent, and regulation of medical assistance in dying in Canada. Welcome.
Thank you for that really warm welcome, and to ISBER and the organizers for having me. I'm sure some of you might know that today is a holiday in Canada, and while it might be slightly unpatriotic of me to say, I'm really glad to be spending Queen Victoria's 199th birthday with you here in Texas. So my talk is fairly limited in scope. I'll set out to give an overview of the growing field of app-mediated health research, also sometimes known as mHealth. And more precisely, I'm going to point out some of the opportunities and challenges facing this rapidly developing field when it comes to informed consent. While there's sometimes a tendency to overstate the promise of mHealth research, I think that it challenges us to think in new ways about relationships between researchers and research participants. And this ultimately might help us to understand better how novel technologies modulate research and work to empower both participants and the general public. So over the next 20 minutes, I'll broadly describe how smartphone applications are part of a shifting research landscape. From there, I'll outline the usual course of app-mediated research and speak generally about the kinds of things such projects usually entail, how they might be of benefit to researchers, and the kinds of unique challenges they might raise. I'll then describe how mHealth research can be seen to impact the informed consent process, and by drawing from examples, we'll work through how the informed consent process might work on a mobile application. And finally, I'll make some comments about the future of mobile health research before concluding. So I'll begin with what's likely to be a fairly uncontroversial observation, that over the last decade or so, smartphones have come to occupy an increasingly prevalent place in our lives. It's likely that few of us in this room don't presently have a smartphone within easy reach. Mine is actually out of easy reach. It's over there, mostly so I don't get distracted by breaking news headlines. So recent estimates put the rate of smartphone ownership among Americans at around 80%. Among Americans aged 18 to 29, smartphone ownership is nearly universal at about 92%. So added to this, about 60% of smartphone users have downloaded an application or multiple applications that have health-related functions. And these functions range from keeping track of physical activity to recording blood glucose data and assisting with prescription management. Yeah, that's... So added to this, smartphones have also become increasingly technologically sophisticated with better sensors, faster processing, vastly increased memory, and a range of functions that would have been really hard to imagine only a few years ago. So it might be unsurprising that against this backdrop, health researchers have taken an interest in mobile applications. And this is so for a really obvious reason. Most of us are just carrying around these really powerful computers that are capable of recording and storing mountains of health-related data. For some commentators, smartphones have really generated a data bounty that's out there for the taking. So with this in mind, ResearchKit was launched in 2015 and ResearchStack followed shortly thereafter. These are open source platforms for the development of app-mediated research projects. They were launched for Apple's iOS and Android platforms, respectively. So the earliest ResearchKit projects studied diseases such as asthma, type 2 diabetes, Parkinson's disease, heart disease, and breast cancer. At present, there are dozens of ongoing research studies led by researchers around the world in such diverse places as the University of Cape Town, the University of Nebraska Medical School, and Freiburg University. In each of these cases, the relevant projects are capable of gathering large amounts of data, and unsurprisingly, the kinds of data varies greatly depending on the study. So here are 3 examples. The My Heart Counts study, which is led by researchers at Stanford Medicine, aims to develop an understanding of the relationship of physical activity and heart disease. It passively collects daily activity records and also actively collects the results from short physical activity tests. And participants may choose to manually input data on things like cholesterol and blood pressure. Concussion Tracker is an exploratory study interested in cognitive function following a concussion diagnosis, and it collects heart rate data and the results of physical and cognitive tests that are also administered by the application. And finally, the Mole Mapper study, which I'll talk about at greater length in a moment, primarily collects photographs of moles that are taken by the participants themselves. So I think we can draw out of this that app-mediated research has several important benefits for researchers. For one thing, research conducted on mobile applications removes many of the geographic barriers that often preclude research participation for certain segments of the population, and especially, for example, from folks who live in rural areas. It's worth noting, though, that there are often country restrictions that prevent participation from outside of the United States. So most research kit studies, for example, aren't available for users of the Canadian App Store. Another clear benefit is the way smartphone apps allow data to be collected and interpreted in real time. So for much of the relevant data collection, there's no need to wait for research subjects to visit a research center in person, which is an obvious advantage. For similar reasons, app-mediated research allows for many more iterations of assessment than traditional kinds of research might. If data needn't be collected in person, for example, the project might be able to expect substantially more interaction on the part of the participant. And finally, app-mediated research has the critically important advantage of permitting real-time and extensive research participant feedback. So most of the studies I've mentioned up to this point give directed feedback, usually in the form of test results, and these test results are often measured against the research cohort or against the general public. But this kind of research faces some distinctive limitations as well. And one obvious limitation are selection biases. For studies available on the ResearchKit platform, for example, participation is necessarily limited to iPhone owners, and I think we know that iPhone owners might not be representative of the public. Perhaps most sort of immediately pressing is the challenge posed by participant retention. So researchers have found that while interest in app-mediated research tends to spike shortly after a study is launched, it often quite precipitously drops off shortly thereafter. So keeping participants engaged and interested in the study is an unresolved and ongoing challenge. Of course, one of the perennial ethical and legal challenges facing any large data set turns on issues related to storage and access. And this is equally the case in the app-mediated research context. So these kinds of projects will need to grapple with questions about maintaining data security and explaining access to participants, while also facing additional worries about the kinds of information that's stored on the participant's smartphone to which the project has access. So to give a tangible example of what I mean, for applications that access a participant's photo stream, we might be concerned about what exactly the software permits researchers to see. These kinds of access concerns link up directly with privacy issues. And from the perspective of app-mediated research participants, there are some really easily discernible ways in which privacy might be raised throughout the process. So one of the most visceral of these is the worry that research apps might be capable of tracking a user's location. So if participants have the impression that an app is continuously obtaining location data, or if it is in fact doing so, we might be worried about the extent to which the participant's privacy is being protected. Now, I think that clearly issues around privacy, data access, and security are some of the most challenging and pressing issues that face research taking place on mobile applications. Smartphones obviously often contain highly sensitive data and information, and finding a balance between encouraging their use in research participation while also safeguarding a reasonable interest in privacy is, I think, going to be a dominant subject of discussion. So I promised that I would say something about consent, and over the next couple of slides, I'll begin outlining how the informed consent process, when conducted over a smartphone, might raise some unique concerns. So, informed consent in the mHealth research context poses some distinctive challenges. For one thing, conveying information to prospective participants might be especially difficult. While the fashionable trend seems to be moving in favor of larger device screens, smartphones are still relatively small, and this, I think, raises some really real questions about how a 30 or 40-page consent document is going to be best shared with the prospective participant. So we know, for example, that participants are really unlikely to want to scroll through a dense, lengthy document on a tiny screen with an often dull or small font. So I'll talk a bit about how most research projects address this concern in a moment. Another challenge is the impossibility of app-mediated research participants interacting contemporaneously with researchers. In some more traditional models of informed consent, a researcher would sit down with a prospective participant face-to-face. This, for most mHealth studies, would sort of defeat the purpose and undermine some of the major advantages of doing this kind of research in the first place. But for the participant, the absence of a real-time interaction means that questions they have might go unanswered, or some of their concerns might not be alleviated. Linking up with the first issue I raised, ensuring that the right balance is struck between user-friendliness and the completeness of the informed consent process is really critical. So what I mean is that while we are aiming to ensure the ease of participating in this kind of research, there should be a similar emphasis placed on ensuring that this doesn't come at the cost of compromising the full disclosure of study characteristics. Further expanding on this, we might want to give some attention to the consistency of informed consent documents throughout this process. Now, most mHealth research projects will use less technical language when communicating with prospective participants, and then also make available privacy policies and consent documents that rely more heavily on legal and medical jargon. And it's sort of crucial to ensure that these documents actually say substantively the same things. For obvious reasons, this would reduce opportunities for disagreement. We might also want to be attentive to the correspondence between documents presented to participants and those that are submitted to an IRB. Obviously, this is a concern in research ethics generally, but it seems especially salient in this context because sometimes in an effort to make things easier to comprehend for a general audience, the sort of audience being targeted by these applications, some nuance in meaning might be lost, and it's necessary to avoid that wherever possible. And finally, oh, did I go too quickly? I think I did. As in most other sort of modern research paradigms, how the informed consent process manages expectations on the return of results and incidental findings is really a central concern. I'll come back to this in a moment. But for now, I just want to briefly run through the informed consent process in practice and try to identify some of the ways mobile health studies address the concerns I've posed. So in doing so, I'll draw primarily on the MoleMapper study, which, as I said, well, I didn't say directly, but I think implied that it primarily aims to gather data for melanoma research. So participants in this study contribute photographs of moles that are usually measured against some object of a standard size, such as a quarter or a dime. This allows participants to track the growth of their moles, if there's any growth, over time. The consent process begins with a simple screening. The sole eligibility criteria is age, so this is a fairly simple process. Of course, we can see right away that this isn't exactly a foolproof way of measuring exclusion. But that's sort of how the process works nonetheless. From there, we can see how MoleMapper addresses some of the difficulties associated with communicating complete consent the complete consent document given the limitations of a small smartphone screen. And it uses something we might call the learn more method. So as you can see, a few introductory points are presented, and prospective participants who'd like to have more information are prompted to follow the Learn More link, where more detailed and technical description is provided. As you can see here, there's a little bit more detail about the way the study functions. So interestingly, this process is usually at the prospective participant's discretion, so they can bypass the more technical language you see on the second screen if they choose. Here's how MoleMapper conceives of the return of results. The study is sort of adamant that it's not offering a medical diagnosis, and the more detailed description in the second screen shows how the application approaches the return of results in a similar way. We might wonder, though, if research participants would conceive of the information being provided by the app in a way that differs from how they'd conceive of basic medical care. That's a larger issue that we might set aside for now, though. So after going through this consent process, the app tests prospective participants on their understanding of the goals and objectives of the study, as well as the obligations researchers have undertaken. So it does this with a series of quiz questions, and about a third of research apps that are currently available use some variation of this technique. The quiz is fairly short on MoleMapper, and once it's correctly completed, the informed consent process is essentially complete. Different apps conceive of this evaluation in different ways. So for some, a prospective participant who fails the evaluation will be prompted to return to the beginning of the informed consent process and begin from scratch. But in other cases, the participants are instead sort of given an opportunity to be corrected about their assumptions or about the way that they answered the quiz, and will be given an opportunity to continue and enroll in the study. So finally, once this is all done, the participant submits their signature, which is signed on the screen, and they're sent to a central system where they are permitted to enroll in the study. So as we can see, this consent process is pretty extensive, but despite its sort of extensiveness, there might still be some challenges. One set of worries are about the form research consent takes in the app-mediated context. So it sort of seems unsettled how best to ensure that participants are satisfactorily informed while participation isn't also discouraged by some sort of cumbersome user interface. And you know, it seems sort of obvious, but the mobile platform might not be ideally suited to ease of comprehension among participants. Another set of worries is about the actual substance of the informed consent itself. So things like ensuring consistency across documents and protecting vulnerable populations sort of remain top of mind. In the MoleMapper study, for example, it isn't clear if there are measures that could be taken to protect the interests of children who might find it easy, sort of practically, to enroll in this study, since the exclusion criteria it's not difficult to work around. So with these issues in mind, we might want to spend a moment looking to the future. So why, from a broader social perspective, does this matter? Well, for one thing, a number of app-mediated studies are designed with the explicit goal of creating tools that are capable of diagnosing disease. So we can see here that both the MoleMapper and My Heart Counts studies, for example, are aimed at testing the possibility of using mobile health apps for eventually diagnosing things like skin cancer and heart disease. And this is part of a broader, more global trend toward increasing automation in both research and in the clinic. So mobile health applications might conceivably play a role in the vastly increased importance of these kinds of technologies in the provision of healthcare in the very near future. So in conclusion, we can note that app-mediated research is proliferating quite rapidly. Nearly anyone with a smartphone can become a research subject with very little effort. And while app-mediated research holds significant promise, the informed consent process presents some unique challenges. As applications come to play a more central role in healthcare and health research, we'll increasingly need to account for these challenges. The digital future of medicine is being constructed in the present, and with mobile health research laying the groundwork for a potentially revolutionized medical practice. So we've discussed some big ideas in this session, but we should try not to forget about the power and promise of the small devices that live in our pockets. So, thank you.
Do we have questions? Come up to the microphones.
Hi, I'll let you sneeze.
Sorry, I was just a little cough.
Sorry, Jane.
Dry, right? Yeah.
Yep. I'm Jane Carpenter from the University of Sydney. I just wondered if you had done, or if you knew, if there had been done any comparative studies about the comprehension of people who are signing up consent on an app versus an old paper-based traditional method, or if those things should be done, if they've not been done.
I think I don't know of any studies that have compared the sort of the capacity of participants to comprehend.
Yeah.
But absolutely, I think that would be interesting. I think at the moment it's sort of just conjecture that paper-based consent is often difficult, so it would stand to reason.
Yeah, and it's hard to know if people do actually understand what they're signing on a paper-based form as well, so it'd be good to do that analysis.
Yeah,
I think.
absolutely. Yeah. Thank you.
Hi, I'm Melissa Reeves from the National Eye Institute. So clearly these apps are useful for the average person, so in the instance of tracking moles. Assumably in relation to possibilities of skin cancer. So my question is, how do you think these apps will be useful on a clinical level? I mean, you're referring people, I think, to go see their providers if, they get whatever. How many people are actually going to do that, maybe, or they're more using this on a personal level? And then from the opposite perspective, how receptive do you think doctors will be in receiving this information when their patient comes in and says, I have skin cancer, look at my mole app? So.
Yeah, so as a non-clinician, I guess I could just offer some thoughts. So you mentioned that these are useful for people on sort of a personal level, and that might not translate into clinical utility. I think that just having some sort of photographic documentation of what's happening with your body might itself be useful for a clinician. So in the mole mapper context, it offers an opportunity to build a paper trail that a clinician might find helpful. I'm not sure if a clinician would find that helpful. Maybe they wouldn't. I suspect that might, depend on the clinician. And yeah, so your second question was about, I don't remember what it was about. Sorry,
I was just thinking from a perspective of health, if you really want these apps to be useful for healthcare, I think you need to have buy-in from the physician perspective as well, because I can tell you, you know, from a personal point of view, going through 23andMe, since we just had that talk, there's a lot of controversy about the results from these types of personalized testing, right? So
Right.
you have to have the bridge between the clinicians and these apps and the patients if you really want them to be effective for healthcare.
Yeah, absolutely. And I mean, I think that sort of the mobile application sort of universe has had some amount of uptake from clinicians. I mean, clinicians use applications to do different things. The worry that someone using something like the MoleMapper application or some other kind of research study application would sort of be led into the rabbit hole of conceiving of something happening to them as a confirmable diagnosis and sort of bring them to the edge and worry about generating increased anxiety among its users, I think is a very present concern, especially with something like the MoleMapper application. Yeah. So.
Thanks.
Hi there, Peter Watson from the BT Cancer Agency, just west of you. Question and then an observation. How well does withdrawal of consent work with somebody who has entered into a research study through this kind of portal? My observation would be, just as one of many kind of examples, about a decade ago, I deleted my LinkedIn account after 3 months of creating one to contact with my niece, and then it didn't become of value, so I deleted it. I'd probably get about, I certainly get a LinkedIn invitation every week ever since, having gone through all the exact steps to formally delete the account. So what happens when somebody tries to withdraw, completes withdrawal, and then what kind of privacy do you then have afterwards, having gone through this portal?
So I think your observation actually leads us to like a really interesting observation about how people in practice withdraw from these kinds of studies. I think that there's some evidence that would suggest that people don't go through the withdrawal process. They just delete the application and then sort of move on with their lives, right? Or they stop using the application,
Or they deleted the application and then when they want to withdraw, they can't remember what that app was.
Yeah, exactly.
Yeah.
So I think the bigger problem, or potentially the greater problem with when it comes to withdrawal, is that people will have a tendency just not to withdraw. So any data that they may have provided the researcher is still there. It's still potentially usable, but they're just not participating in the research project anymore. And in practice, the majority of people who leave these studies do exactly what we've just described. Though there are formal withdrawal processes. You will have to log on to your account and state that you wish to withdraw from the study. But in practice, it doesn't seem like that's how the majority of people who leave the study actually approach withdrawal.
Thanks.
Hi, I'm Wayne from the Ingham Institute. I do have 2 questions. The first one is there the apps to be used for consenting for patients for biobanking? And the second question is, what do you think about linking the clinical data or the medical records with the clinical apps research?
So on your first question, I'm embarrassingly unsure, and the first person named on my acknowledgement slide, Manzawati, who I work with, would be a phenomenal resource for those sorts of questions. I'm just not well-versed enough on biobanking to comment. Can you remind me what your second question was? I have apparently a short memory.
Oh, yeah. So I'm asking, what do you think about the linking the medical records to the apps for the research when the patients consented?
Well, so I think that the sort of some of the theoretical consent problems that I've identified are effectively present in the same way, though with the notable exception that a live interaction with a researcher might be more possible in those kinds of situations. A research participant would have a greater capacity to speak to someone in real time. And I think that that's sort of an important thing when we're thinking about the ethics of procuring research consent. Just having a person to talk to and ask questions helps to significantly increase the informativeness of that consent. Thanks.
Okay. We thank you very much. Thanks.